People taking Ozempic for weight loss started reporting something nobody designed for. Their alcohol cravings disappeared. Their urge to smoke faded. The compulsive pull toward gambling, online shopping, mindless scrolling, quieter. They were not trying to quit any of those things. They just stopped wanting them. This happened consistently enough across thousands of patients that researchers began asking an uncomfortable question. What if Ozempic is not really a weight loss drug? What if the weight loss is just one consequence of something happening much deeper, inside your brain? The answer involves a set of receptors most people have never heard of, and a hormone your body already makes every single time you eat. Ozempic is the brand name for semaglutide, a drug originally developed to treat type 2 diabetes. It works by mimicking a hormone called GLP-1, glucagon-like peptide 1. Your gut produces GLP-1 naturally after every meal. It tells your pancreas to release insulin. It tells your liver to slow down glucose production. And it sends a signal to your brain that you have had enough. Semaglutide does all of those same things. But where your natural GLP-1 breaks down within minutes, semaglutide stays active for a full week. Think of it as the same key, but one that holds the lock open long after your natural version has worn off. In trials published in the New England Journal of Medicine, semaglutide reduced body weight by an average of 15%. Some participants lost over 20%. Nothing in the history of obesity medicine had come close. And the mechanism, as most people understood it, seems simple enough. Ozempic suppresses appetite. You eat less. You lose weight. That explanation is true, but it is incomplete. And the part that is missing explains everything. The food noise, the cross-addiction effects, and why some people describe taking Ozempic as the first time in their lives they have ever felt genuinely free from food. GLP-1 receptors exist throughout the body, in the gut, in the pancreas, in the heart. But the ones that matter for what you are about to understand are in the brain. Semaglutide crosses into the brain and binds to GLP-1 receptors in at least 5 regions of the hypothalamus, the part of your brain that governs hunger, energy balance, and the feeling of fullness. Each of those receptors is a lock. Semaglutide is the key. When it fits in, it turns down the hunger signal. Not gradually, reliably, for days at a time. But the hypothalamus is only part of the story. GLP-1 receptors also sit in the brainstem, specifically in a region called the nucleus tractus solitarius, which processes signals arriving from your gut and tells your brain whether you are full or still running low. And here is where it gets counterintuitive. They also sit in the ventral tegmental area, the VTA. This is the brain's primary reward and motivation center, the source of dopamine, the region that makes food feel urgent, pleasurable, and worth pursuing. When semaglutide keys into receptors in the VTA, something unexpected happens. A 2025 study found that GLP-1 receptor activation simultaneously reduces appetite and increases dopamine reward signaling in connected brain regions. The urgency of food is turned down. The brain's broader capacity for reward is preserved, or in some cases, enhanced. What people experience from the inside is what they have started calling food noise. The constant mental loop about what to eat next, when to eat, what is in the fridge, whether you deserve something after a hard day. On semaglutide, that loop goes quiet. Not because the person is suppressing it through willpower, because the signal driving it has been turned down at its source. And because the VTA governs reward-seeking behavior in general, not just hunger, the quieting effect appears to extend to alcohol, nicotine, and other compulsive patterns. Ozempic is not selectively targeting your cravings for pizza. It is resetting the volume on the brain's entire reward system. The food just happens to be the first thing most people notice. If you watch the first video on this channel, you already understand what happens downstream when this system works correctly. Eating less means a smaller glucose spike. A smaller glucose spike means a smaller insulin response. And as that video explained, lower insulin is precisely what allows the fat storage lock to begin releasing. The brain signal at the top and the metabolic system below it are not separate stories. Change the input to one, and the other shifts with it. Here is what matters if you are not taking semaglutide and have no intention of starting. Your body already makes GLP-1. The question is what triggers it to release, and what is preventing you from getting enough of it. GLP-1 is produced by specialized cells lining your small intestine and colon. They release it in response to food, but not all food triggers the same response. Protein is one of the strongest natural stimulants of GLP-1 secretion. Starting a meal with protein, eggs, fish, Greek yogurt, legumes, before the carbohydrates triggers a larger initial GLP-1 response. A stronger satiety signal reaches the brain before you have eaten the bulk of the meal. Fermentable fiber works through a different route. Foods like oats, cooked and cooled potatoes, green bananas, and beans feed the bacteria in your colon. Those bacteria produce short-chain fatty acids, particularly one called butyrate, which directly stimulate GLP-1 release hours after the meal has finished. You get a second wave of satiety signaling from the same food, long after digestion. A 10-minute walk after eating has been shown across multiple peer-reviewed studies, including research published in Diabetes Care, to meaningfully reduce the post-meal glucose spike. Less glucose spike means less insulin, and the same post-meal activity has been shown to enhance GLP-1 activity in the gut, giving the body an additional satiety signal at exactly the moment it is most useful. Sleep is the factor most people underestimate. GLP-1 receptor sensitivity is measurably higher after adequate sleep. A single night of poor sleep reduces the brain's ability to respond to GLP-1 signals the following day. The same meal leaves you less satisfied. The same food noise is louder, not because of what you ate, because of what you did not do the night before. None of these are substitutes for a week of continuous semaglutide receptor activation, but they are working on the same system, the same locks, the same keys. Ozempic works because GLP-1 receptors inside your brain, not just your digestive system, are what govern your relationship with food, reward, and craving. Semaglutide is a key engineered to hold those locks open for a full week. The result is not simply eating less. It is a quieting of the hunger signal at its origin point, and a resetting of the reward system that makes food feel so urgent in the first place. Your body has this system. It is running right now. The question is how well you are supporting the inputs it was built to respond to. Protein before carbohydrates, fermentable fiber, a short walk after eating, enough sleep. These are not hacks. These are the signals the system was designed to receive. The food noise was never a personality trait. It was always a signal. And signals, with the right inputs, can be changed.